Hello everyone. I welcome the students, scholars, researchers, and the scientists all over the world. This is Advanced Multiple YouTube channel. Advanced Multiple Incorporation actually a knowledge processing and information management organization that is located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir. I'm working as scientific executive with Intellectual Consortium of Drug Discovery and Technology Development in Corporation. Okay, well, today I'm going to share my knowledge, my experience, and my research under the title of Classification, Source, and Function of Microbial Enzyme. For this purpose, I collected the information and scientific data from different sources like PubMed, Medline, Scopus, Google Scholar, National Library of Medicine, MBAS, Directory of Open Access Journal, ProQuest, CNKI, that stands for the China National Knowledge Infrastructure, that is China database company provide the scientific and research information all over the world. Some of the information collected from Crossref and the ResearchGate. Okay, well, before moving forward, I would like to talk a little bit about microbial enzymes. Enzymes are biocatalysts or the chemicals produced by living cells to bring about the specific biochemical reaction, generally forming part of the metabolic process of the cell. All enzymes which have been purified are protein in nature and may or may not possess a non-protein prosthetic group. Enzymes are highly specific in their action and substrate, and often many different enzymes are required to bring about by concerted action the sequence of the metabolic reaction performed by the living cell. Okay, well, there are the four different type of the microbial enzymes like bacterial enzyme, viral enzyme, fungal enzyme, and yeast. When we divide something or some entities, we have to design some criteria. Okay, we use the criteria here for the classification of the enzyme is on behalf of the creatures. The bacterial enzyme they call as bacterial enzyme, then viral enzyme, and fungal enzyme, and yeast enzyme. They are produced by the four different category of the organism, four different creatures on the earth. Okay, well, I'm going to talk a little bit about the enzyme, its source, and its functionality, its role in the bacterial life. The first enzyme produced is amylase, that is synthesized by the bacillus. Bacillus is one of the bacteria that belong to the gram-positive uh, gram-positive rod, and these bacteria most of the time anaerobic. They don't need oxygen for their metabolism, reproduction, and to live for a long time on the earth or like in his life. What is the function of the amylase? Amylase used to convert the starch into the sugar. Second enzyme is the gluconase, that also produced by the same category of the bacteria bacillus, and it is used for the cleave of glucose. The third one is the glucose isomerase, produced by the same type of the bacteria, and that catalyze glucose to its isomeric form. The third type of the bacterial enzyme is hemicellulose, produced by the bacillus, and it catalyzes the hydrolysis of hemicellulose. The next type of the enzyme is protease, produced by the bacillus, same bacteria. So it actually do catalyze split of the protein. The last one is the Fullanase, that is produced by the Klebsiella, that is one of the type of the bacteria belong to the gram negative. Their shape is rod, and most of these bacteria are anaerobic. They catalyze and hydrolyze the polysaccharides such as pullulan. There are also many other enzymes produced by different kind of the bacteria, but this is the like exemplary or major type of the enzyme that produce or synthesized by the different bacteria. Okay, the second type or second category of enzyme, they belong to the fungi called as fungal enzyme, and I'm going to talk about the name of enzyme, its source, and the function or the operation for what purpose that is used in fungal life. Amylase produced by the Aspergillus that convert this starch to the sugar. Catalase produced by the Aspergillus, same type of the fungi. It used for the oxidative catalyst, decomposes hydrogen peroxide into the oxygen and water. Cellulose, that is produced by the trichoderma, and it used for the catalysis of this cellulose. Dextranase, that is produced by the penicillium, and it is used for the catalysis of the dextran. Lactase enzyme, that is produced by Aspergillus, and it did catalyze the lactose. The next enzyme is the lipase. Lipase produced by the rhizopus, and it is used for the purpose of emulsifying or emulsifies the fats. Then the same category of the enzyme continue rennet, and that is one of the enzymes that is produced by the mucor mihi that did curdle convert the curdle milk to cheese and junket milk dessert. Pectinase is produced by the Aspergillus that catalyze the pectin. Pectin lyase, Aspergillus, that is the fungi produced, and it catalyzes the pectin. Same function, same role. Protease derived from the protein. It disintegrates the protein produced by the Aspergillus. Then raffinase that is produced by the mozzarella. It catalyzes the raffinose, a trisaccharide found in the sugar beet, cottonseed, and certain other cereals. Okay, next type of the enzyme belong to the yeast, and as usual, we are going to talk the type of the enzyme, its source and functionality. Lactase that is produced by the Cloviromyces, and it catalyzes the lactose. Invertase produced by the Saccharomyces that catalyze the hydrolysis of sucrose into the glucose and fructose. The lipase that produced by the Candida emulsifies fat, so the fat. Raffinase, Saccharomyces, that is the yeast produce, and it actually catalyzes the raffinose. Okay, well, in conclusion, microbial cells used for the production of the enzyme on industrial scale. These enzymes are obtained from different microorganisms and used in food, medicine, and chemical industry. The industrial use of commercial scale has potentially enhanced the scientific value of these enzymes. Therefore, the catalytic reactions are used in biocatalysis and immobilize enzyme and other specific application in science or in research or on different businesses. Okay, well, before closing my video, I would like to salutate and greet my friends, my colleague, my coworker, Sherry Bachkat, then Wolfgang, Daniel, Dustin Hawker, Marwa Al Maghribi. Then there is Terry Mill, Faisal, Noreen, Kunza, Robert, Amin, Arslan, and Ishtiaq. They helped me a lot to make this video today. I also like to acknowledge the kind help and support of Advanced Multiple Incorporation and Intellectual Consortium of Drug Discovery and Technology Development. And thank you very much for watching the video and staying with me for so long time. I hope I will see you in my next video. Take care. Bye, please.